Good morning to all of you. I am recording this lecture on chimeric protein. It was requested by one of your classmate. So I will focus on chimeric protein in this lecture, as we have already studied chimeric gene in the class, and I hope you keep your notes with you. So I will focus on chimeric protein in this lecture only. What is chimeric protein? Chimeric protein are also called as fusion protein. Means the protein that has been generated because of combination of two or more genes. So these chimeric proteins are also called as hybrid protein because they contain one domain from one protein and another domain from another protein. So the resultant protein is known as hybrid protein. It may have This protein may have different function. This protein may have different physiological properties, physiochemical properties. Say for example, a protein needs to be folded like this in normal condition, but under as we have made it a chimeric protein, so it may not fold it like this. It may fold this way. It may either choose to not to be folded. So due to this physiochemical changes, due to change in structure, due to replacement of domain, the protein may have different physiochemical patterns. It may have loss of function, it may have gain of function. Chimeric protein is not just that they are synthesized in vitro, they are also produced in vivo. In vivo, the chimeric proteins are result of some mutations. Complex mutations result in formation of chimeric protein. These mutations can be chromosomal translocation. Say for this gene has two parts, A and B, and this gene has three parts, A, B and C, and this C part replicate itself and translocate to gene A. Okay, then this is called as chromosomal translocation. So the resultant protein that will be produced now will have domains of gene 1 and will have domain of gene 2. It may have gain the function or it may lose the function. Same way, the chimeric protein can be produced by duplication, where one segment of DNA has been duplicated and translocated some other position. So the resultant gene become a chimeric gene and the protein will become chimeric protein. Retrotransposons, as we all know that genome has some transposons, of which some are retrotransposons. These retrotransposon has specialty that they replicate by reverse transcription, and this gene is located some other position in the genome, and due to its translocation there become a chimeric gene, and this chimeric gene will produce chimeric protein. The example of naturally occurring chimeric protein is BCR-ABI. It is a fusion gene, and it is result of cancer, and it cause chronic myelogenous leukemia. What a chimeric protein actually is. Say for example, a protein has two domain, A domain and B domain. And in chimeric protein, we will be having the A domain from human and B domain from mice or some other organism. And these two domains are linked with a linker protein. And the resultant protein, which has A and B domain that are linked through a linker domain, are called as chimeric protein. So biologically, chimeric protein can be functional or non-functional. Functional chimeric protein usually have all the functional domain of a gene, but they have regulatory genes that are added through in vitro gene transformation or through in vivo gene transformation. So due to addition of regulatory genes, these genes may become upregulated upregulated or downregulated. When downregulated, they their function become less, means they have to produce, say for example, an amount of protein, but now they are producing n minus 1 or so. They are producing low amount of gene, or they are producing dysfunctional protein, so they are down regulated. If they And in upregulated situation, this situation is totally opposite to this, where the protein become more functional. Protein shows more variation in the function, and they have gained the function. Non-functional chimeric protein, they having different functional domain. As we have seen in previous example, we have one domain from human, one domain from mice. So they have a different functional domain from different organisms that are linked together, and in this condition the chimeric protein most probably will lose its function, so they become non-functional chimeric protein. Okay? And due to this, due to as they have different domain from different organism, they tends to misfold, or they altogether do not fold. And if a protein do not fold, they will lose its function, they become non-functional. A chimeric protein can be an fusion protein or C fusion protein. As we all know that a protein has two terminal, N terminal and C terminal. So a chimeric protein may have a fusion protein that has fused to its N terminal, or it may have the fusion protein that has fused to its C terminal. Say for example, we have a protein P1 and another P2. They are performing same function in different organisms. If we fuse these protein with each other, they may result an fusion protein, where N terminal of P1 is retained and C terminal is fused. So this becomes C terminal protein. And if N terminal is replaced by protein P2 and the C terminal of the uh, actual protein is remain same, so uh, it become a protein that has N terminal fusion protein. So this was C terminal fusion, and this was N terminal fusion. So it can be understood in different ways. Say for example, a protein has two domain A and B. This the same protein in different organism also has two domains A and B, and these two domains are fused. We may fuse A of human and B of mice, or B of human and A of mice. So the resultant both of these protein will be chimeric protein. Why do we produce chimeric protein? The question is, why do we actually need chimeric protein, or what is the intention to produce chimeric protein? The We produce chimeric protein for different intention. Say for example, we want to increase secretion of a protein, so we may produce a protein that has secretion ability. It is mainly done in case of antibodies. So we all know that antibodies are two type, fused and secretory. So secretory antibody may have the secretory domain of mice, so as to increase its secretion. Solubility, the protein to increase solubility of a protein, so and that it has some domains that increase its solubility in plasma. Or stability, that we produce protein to increase its stability, or we may produce the protein to increase or decrease its function. As we know, for production of chimeric protein, we must need some its expression. So if a protein is to be produced for human use, it must have eukaryotic expression system. In, as we know, in eukaryotes, gene has a promoter, it has a inducer, and it has a structural gene. All three components must be intact if we want that a protein should be expressed in human. Further, as we know, human are eukaryotic. So in eukaryotes, there must be a leader sequence so that the protein is transferred to some organelle where it is required, so that protein is tagged to Golgi bodies if it is to be secreted, so that a protein is tagged to endoplasmic reticulum where it has to be folded in proper way. For this, we need to add a leader sequence. Another important thing at the level of transcription taken in consideration is addition of cap and tail. As we know, mRNA of eukaryote has a cap and tail so that it is transported outside the nucleus where it is to be modified, it is to be tagged with cap and tail so that it can be easily transported outside the nucleus, as protein synthesis in eukaryotes occur in cytoplasm. Further, if we have taken a gene from lower organism, say for example we have taken a gene, bacterial gene, to synthesize human chimeric protein, and as we know that we want to synthesize this human gene inside a bacteria, or we want to synthesize a prokaryotic gene in human. So there must be codon optimization. Codon optimization, what is this? That a codon, if it is synthesizing, say for example AUG, if it is coding for methionine in bacteria, it must code for methionine in bacteria. If it must code for methionine in human. If this is not the case, then our reason of producing chimeric protein will wear off. We can't produce functional protein. If a code that codes for some amino acid in bacteria and codes some other amino acid in human, so there is chances of loss of function. So we must choose the code that are same for human as well as for bacteria. It is structural of a chimeric protein for thyroxine hormone. As we know, it must have an inducer, it must have a pro ASP site which is a cleavage site, and his tag which is used for purification, and a structural gene. This structural gene is used for its expression for thyroxine hormone in human. It must be having inducer, it must be having pro SP site for its cleavage, and it must be having his tag for its purification and the structural gene. This all gene must be under some promoter if we want to it to be expressed in all cell. We must having constitutional promoter. If we want to it must be expressed in different specific cell, it must be having a specific promoter. Now, how can we produce a chimeric protein? What are steps taken for the production of chimeric protein? Uh, First of all, we must select a gene. We must select a gene from gene library. We may select it from cDNA library. And if we have selected the gene, we we do blast studies so as we can identify which of the two proteins we can fuse, so as we can gain a functional chimeric protein. Of these two protein, of these studies, we identify the gene and we make chimeric gene, and from this chimeric gene we produce chimeric protein. We have already studied in previous class how we produce chimeric gene. I have told you about uh, random gene combination and Rachit, the two techniques I have already taught you. And now, if we have identified the gene, we say for example protein has two domain A and B, and for this we have two genes A and B. These gene segments are restricted, digested so as to produce complementary sites. These complementary sites are that they can fuse with each other when required, and we may use a joining or linker segment uh, to successfully join these segment. Out of these two genes, we may construct a vector. This vector can be a bacterial vector, it may be a plasmid, it may be a cosmate, it may be a basimate. So a vector, this vector must have, as we all know, it must have an origin of replication. It must have a lac1 gene and an marker gene for ampicillin resistant, and the structural gene which I have described in previous slide, that it must be having a promoter, it must be having a inducer, pro ASP site, his tag, and the structural gene. It must be under a T7 constitutive promoter. This vector is induced in bacteria as binary vector or a single vector. This is single vector in which both the gene are inserted in a single vector. A binary vector which may be having two different genes that are inserted into two vectors which have the two different genes, and they express inside the bacterium and link together when they are produced in the bacteria. So these are binary vector. When a gene is induced in a host cell, say for example we have induced this gene in E. coli, it will produce mRNA which must be having a leader sequence and a protein sequence. The leader sequence is to be digested, it is to be broken down, and the protein must be folded in a way the eukaryotic protein folds so as it become functional. Other method of producing functional protein is through baculo baculovirus expression vector. Baculovirus expression vector must be having circular DNA which has a leader sequence, which has left border and right border sequence which are denoted as L and R. Then we must be having a promoter, then gene of interest, and a terminator gene. This baculovirus gene which we have synthesized is crossed with nascent baculovirus gene which having left border and right border sequence, and polyhydrin gene. This polyhydrin gene is to be replaced with our gene of interest in this crossing over. So the resultant vector which is synthesized must be having left border, right border, terminator, and gene of interest, and this gene of interest is inserted into baculovirus so as to produce our chimeric gene. We may also insert this vector into insect or E. coli so as to produce the chimeric protein. The modification for insect is that this baculovirus expression vector is converted into base backmid, which has bacterial origin of replication and a MCS, that is multicloning site, and antibiotic resistant gene. So these gene can express itself in E. coli as well as in insect. That's all for chimeric protein. We may add a utility of chimeric protein, which I have already uh, taught. I have in I have added in this video as I started this. So thank you.